I will talk about my master degree project, genetic engineering of seeds to enhance their radiation resistance for long-term space missions. Out there in space, there is a plethora of space stress factors like low temperatures, hypoxia and hypoxia conditions, hyperbaric conditions, microgravity, and ionizing radiation. And specifically, ionizing radiation is the most lethal factor to living organisms. But at first step, we need to understand what is radiation. Radiation is a physical phenomenon that consists on the emission and propagation of energy through materials and through space. And the energy can travel in form of electromagnetic waves like visible light, UV light, X-rays, gamma rays, and in form of subatomic particles like alpha particles or beta particles. And well, both types of radiation affect living organisms differently. And the degree of the damage also depends on the level of the energy they possess. So we understand radiation can affect living organisms in both ways if it is electromagnetic waves or subatomic particles. In space, we have two main sources of radiation. And the first one and the most evident is the sun. Sun provides Earth with electromagnetic radiation necessary to life, but mass ejection coronal even produce high energy and high speed protons called solar particle events. And the other source of radiation in space are supernovas explosions, which produce complex mix of electromagnetic waves and subatomic particles. And this kind of radiation we call galactic cosmic rays, and it is composed by protons, atomic nucleus, alpha particles, and heavy ions. And as you can see in space, there is a complex mix of radiation, so lethal for us. But I have good news and bad news. Good news, this kind of radiation can affect living organisms on Earth because we have two strong blind shields. First one is atmosphere, because atmosphere gases retains radiation, generating secondary particles and secondary molecules. And the other shield, most powerful we have, is a magnetosphere, which protects Earth to this space radiation. But if we talk about long space missions, that means the spacecrafts will be so far away with this protection, and any biological sample inside will be exposed to deep space stress factors. Scientists believe this radiation can get through materials, but in 2013, the measurement of energetic particle radiation in transit on Mars when rover Curiosity were sent, revealed that both galactic cosmic rays and solar particle events not only could get through the spacecraft materials, even form secondary particles, most dangerous than the first source of radiation. That's because NASA creates a research group dedicated to identify technological and biological challenges to this kind of missions. In 2024, they exposed that the protection of dry seeds in long-term space missions, it's still an unsolved problem. And to understand how galactic cosmic rays affect seeds, in 2018, Califar et al. Did a little experiment. They set two groups of Arabidopsis thaliana seeds under laboratory conditions, radiation laboratory conditions, and in situ conditions above Antarctic stratosphere. So here, in left panel, we can see how germination rate decrease in the case of laboratory conditions radiation and in case of Antarctic radiation. In both cases, germination rate decrease. And in the right panel, you can see how mutation rate increase, even in both cases. On the other hand, transcriptomic analysis in irradiated seedlings of the same model plant reveals about some 7 years involved in sensing of damage, DNA repair, and cellular defense that has or present expression levels directly proportional to radiation doses. As you can see in red, the expression level of that genes at 40 centigrades, and in green, you can see the level expressions at 80 centigrades. In general, radiation can induce somatic mutations on plants, heritable mutations on sexual organs like ovules and pollen, increase susceptibility to diseases, decrease germination rate, induce both premature germination and premature aging, produce seed death, and even negative effects on palatability. Well, at molecular level, radiation can damage DNA in both ways, directly and indirectly. In direct way, radiation can interact with hydrogen bonds and break them, and even can interact with phosphate change and break it. And in indirect way, radiation can interact with water molecules, ionizing or breaking it, generating other kind of molecules, very tiny but very reactive, called reactive oxygen species. And these molecules are very dangerous to the cells because produce lipids oxidations. That means that these molecules can break the membranes of the cells and cause lipids oxidations, proteins oxidations, and more punctually produce damage in DNA. Single strand breaks, double strand breaks, intramolecular and intermolecular DNA crosslinks, change of bases, iteration product, loss of bases, pyrimidine dimers, and bridging with other kind of proteins. In nature, we have some organisms very resistant to radiation. Here are some of them. Thermococcus gamma tolerans, it's an archaea which resists 30,000 of rays. Deinococcus radiodurans, it's a bacteria which resists 15,000 rays. And Ramacetus varionatus, it's a tardigrade which resists 4,000 rays. And Syntriche caninervis, desert mouse, which resists 5,000 rays. If we compare these organisms with the doses which resist humans and common plants between 8-9 grays and common plants between 50 to 200 grays, well, we can see these organisms are highly resistant, and they can do it because they have efficient DNA repair mechanisms in matter of fidelity quantity, speed, and other kind protective proteins. And that's why my hypothesis consists on the transgenic induction of genes encoding protecting proteins from radiotolerant organisms, such as Deinococcus radiodurans and Rhamnosotius varionatus, could protect plant embryos to cope with DNA damage from galactic cosmic rays in deep space. My main objective is evaluate the protective effect to heterologous expressions of the D, P, and A proteins from Deinococcus radiodurans and Rhamnosotius varionatus in Arabidopsis thaliana seeds. And I have two specific objectives. First one, to generate Arabidopsis transgenic lines, and the second is evaluate the ability of heterologous proteins to mitigate effect of oxidative stress on embryos cells. Well, this is my molecular strategy. I choose two model extremophile organisms, Rhamnosotius varionatus, a tardigrade, and Deinococcus radiodurans, a bacteria. Then I choose three the best three genes of these organisms. And the first one is the protein of tardigrades, an intrinsically disordered protein which protects DNA at chromatin level. That's because protein, the N-terminal side of protein has affinity with nucleosomes by electromagnetic charges differences. And this protein creates a micro or tiny electromagnetic field capable to divert electromagnetic radiation and avoid the interaction of reactive oxygen species with the DNA. The other protein is P protein from the Deinococcus radiodurans, and it's a master regulator. The protein senses the single strand break so quickly. That's because the protein is activated by the direct incidence of radiation. And in its host, the bacteria, protein even can regulate other kind of genes involved in oxidative stress by radiation and by desiccation. And the heterologous expression of this protein in plant cells works. But scientists still don't know how nor why, but works. And the last protein is a protein from Deinococcus even. And this protein is a nucleoprotein, and it has wonderful mechanisms because protein sensing double strand breaks repair so quickly, and after that, it begins to polymerize itself along DNA change. This kind of adhesive molecular adhesive tape has two proposes. The first one is to avoid the enter of endonucleases to disintegrate DNA, and the other mechanism is form complexes with ligases to stimulate the efficiently DNA repair. So it's amazing protein. Well, the main idea is to express these three proteins on quiescent states of plants and then result a health seedlings at the end of the transit in space. I achieved this choosing two kind of promoters. And the first I chose was a specific promoter of abscisic acid insensitive 3 or AB3. And this promoter expressed my proteins only in an embryo cells, endosperm cells, and seed coat cells. As you can see in red, the high-level expression of this promoter is only in siliqual stages or embryo stages. And the other promoter I chose was a constitutive promoter, the 35S cauliflower mosaic virus, which expressed my proteins in all the tissues of the plant and in all the development stages. Well, I did the design and construction of all the vector expressions. Then I synthesize the specific tissue expression vectors, and then I did the construction of constitutive expression vectors by molecular cloning, first in Escherichia coli, then in Agrobacterium tumefaciens, and then I conduced the transformation of Arabidopsis by floral dip technique. And that's the way I select transgenic seedlings. In the left, you can see a plant resistant to ioromycin B, the antibiotic of resistance of AB3 constructions. And in the right, you can see a plant resistant, a seedling resistant to kanamycin, the antibiotic resistance of constitutive constructions. Okay, so I generate 4 transgenic lines of the protein fused to AB3 promoter. In the right side, you can see a photography of all my transgenic lines. And in the right side, you can see the amplification of genes, resistance cassettes, and GFP, the fluorescent protein, by PCR. Well, then I generate other 5 transgenic lines of the D protein fused to constitutive promoter. In the case of P protein, I generate 4 transgenic lines fused to AB3 promoter and other 4 transgenic lines fused to constitutive promoter. In the case of protein A, I generate only 3 transgenic lines fused to AB3 promoter and only 2 transgenic lines fused to constitutive promoter. These results for these cells can show us that maybe the protein could be involved in a deleterious effect, a penalty, a fitness penalty, because I generate less transgenic lines in contrast with the other 2 proteins. And currently, for this protein, I detected the protein by Western blot in a mix of seeds generated by AB3 promoter plants, and detects the protein in the leaves of the two transgenic lines with the constitutive promoter. And even I detected the protein in Nicotiana tabacum bentamiana and Nicotiana bentamiana leaves, and I will explain why. That's because moreover, I did the transient expression of my proteins on tobacco. That's because I wanted to prove their biological activity. So after transformation, I put the plants below UVC radiation stress. And here you can see how wild type plants dead after 5 days of exposure. And obviously, after 15 days of exposure, they were completely dead. And in the case of the plants transformed with the three proteins, after 5 days of exposure of radiation, they are still alive. And after 15 days of exposure, they not only alive, but also are recuperated. Currently, my transgenic seeds are at Institute for Nuclear Science being irradiating with gamma rays. But meanwhile, I'm been analyzing the behavior of wild type seeds under gamma rays. So I choose for doses of radiation uh, 1,000 grays, 1,500 grays, 2,250 grays, and 2,500 grays. In the left, you can see how at the lower dose of radiation, mutation rate increase, mortality rate increase, and survival rate increase. And these effects are severe uh, uh, when I irradiated with a high dose of radiation, as you can see in 2,500 grays. Well, in general, after 4 days of germination, in compare with a health seedling, I observed these phenotypes: cotyledon malformation, um, chlorosis in seeds, a lack of root development, lack of root development, and an hyperpigmentation due to increase of anthocyanins. Anthocyanins, it's a pigment involved in oxidative stress. So the effects of radiation are very clearly in wild type. So I hope my transgenic seeds can mitigate these effects or completely separated. Up to here, my conclusions are that the proteins from extremophilic organisms were not harmful to the heterologous plant systems. However, expression or overexpression of protein A could be related to interference in cell cycle. I have two future prospects. If proteins are shown to have protective effect on Arabidopsis thaliana seeds, they could be applied to crops of agricultural importance. On the other hand, if protection against electromagnetic radiation is effective, in this case, gamma rays, well, then their biological activity on the radiation their biological activity were proved on the radiation produced by a particle beam. Finally, I want to thank all my collaborators, Dr. Alicia Negron and Dr. Claudia Fuentes from Institute of Nuclear Science of UNAM, Dr. Elena Kravchenko from Joint Institute for Nuclear Research, Dr. Isai Narumi from Toyo University of Japan, and Dr. Katsuya Sato from Takasaki Institute for Advanced Quantum Science from Japan even, and of course my thesis advisor, Dr. Jean-Paul Fryer from Center of Research and Advanced Studies of the National Polytechnic Institute. I would also like to mention that given my academic background and my interests, I'm eager to continue my continue working on research areas related research areas, molecular cloning or radiobiology at doctoral level. So therefore, I really appreciate any recommendations for laboratories where I could pursue a PhD. You can contact me by email or ResearchGate, and that will be all. Thank you for your attention and thanks for having me.
Thank you for having me. My name is Ismael Acosta. I am from Uruguay. I work at the Laboratory of Biophysics and System Biology of the Faculty of Sciences in the University of the Republic. The overall talk, I would introduce myself a little bit, then I will talk about some experiences, as I put it in the title, on astrobiology in Uruguay from three different perspectives, geology, astronomy, biology, and then I will finish with some outreach activities that I carried out last couple of years as a high school teacher and science high school teacher in Uruguay as well. So this is me. This photo was taken last year on Iceland, where I went to see the Northern Lights. And this to the right is the Faculty of Sciences. I should emphasize that here Uruguay is a small country. We only have 3 million people on all the country, and we only have one university, which is public, and it's the only university which you can pursue a STEM career, which is also very important because as an underdeveloped country, it's important that all the population can have a proper education. So
mute your microphones, please. Sorry, Ismael.
It's okay. Okay. So as you can think, astrobiology is a very interdisciplinary subject. And I shall prove it to you that you can approach it with from different subjects, such as astronomy, biology, or geology. And coming from a country that does not have a history of astrobiology research, you take what you can get, as you can say it. So, okay, let me see. Okay. My first attempt to get introduced in astrobiology was from a geological perspective. As part of my Bachelor in Biological Sciences in 2023, I start studying the evolution of early life, studying some ancient rocks we have in Uruguay of about 2 billion years of age, which is marked here in this chronostratigraphic chart. In fact, this stratigraphic chart is very deceiving because the part I am putting in red with a black arrow represents more or less the half of the age of the Earth, which is 4.5 billion years. So in fact, the age I was studying in this work represents more or less half the age of the Earth. So in Uruguay we have some old rocks. Some of the oldest rocks you can find in South America. In fact, these rocks represent, as far as we know, rocks with organic matter, which are preserved enough to have some fossils with which you can classify them. Okay, so previous work have dated these rocks at around 2.15 billion years, putting it in the Paleoproterozoic era. And the organic matter that is contained in these rocks contains some fossils that can be classified, or at least classified in a morphological way, not in a taxonomical way. And we can study how these morphological fossils could have lived during that time. For example, one of the best fossils we found was this spheroidal fossil, which in the parallel taxonomy of in the literature, they refer them as to Leospheridea. But also we found other types of fossils. I should emphasize that given that these rocks are pretty old, they are get the organic matter gets carbonized due to high pressure and temperature. So the organic matter appears this black. So it is very difficult to assess a morphology, and therefore, if it is biological in nature at all. In this example, we found that there was some colonial strategy of living for these bacteria. We presume they are bacteria because at that time the only thing that it was cyanobacteria and some archaea maybe. But in morphology, they resemble modern algae, which are, of course, eukaryotes. So that put us to think that given that there were no predators at that time, maybe cyanobacteria were exploring all kinds of modes of living. For example, in this case, we have a pyramidal form of living, which is classified into this parallel species, that we can compare morphologically to other newer, younger species. For example, with this 1 billion year old species and modern algae such as Tetrodon that lives in the ocean. So this was my first attempt to study evolution of life. I was interested in how to find ancient life on Mars, how can life get preserved on rocks, how can we study it, how can we assess the biogenicity of organic matter, if we can attribute biological origin. So then I started working on other things. I graduated from my bachelor's degree in biological sciences and get interested in astronomy. And one of my first attempts, I am currently finishing my bachelor's degree in astronomy as well. One of the first attempts in astronomy was studying planetary defense. You would agree with me that planetary defense is a very important topic. The extinction of life is part of astrobiology as well. And in this work, we work with data from the Meteoritical Bulletin database, which records every meteorite fall that comes to Earth, or at least that humans can register. And we have recordings from the 19th century, sorry, and we can put them in a chart and see what the data can tell us about it. So as we can see in the left panel, we see that the number of total falls is increasing, probably due to the increasing of population and more people are registering them. We can have more data, but also the number of damaging falls is also increasing. We are defining a damaging fall as any meteorite fall that has some consequence on human-related activities, such as outcrops, such as cars, buildings, street, ships, etc. And this only suggests that humans are, in fact, populating the whole Earth, and we have more and more people to suffer from meteorite falls. But one of the things that we were interested in was to see if there is a particular moment or a particular position of the Earth in its orbit that could have more meteorite falls than any other time or moment in the Earth's orbit. So in the right panel, you see that the number of falls per solar longitude, degree of solar longitude, which you can take it as a certain position of the Earth in its orbit, or a certain date of the year. In fact, you can see that the data fits properly to a Poisson distribution, telling us that it is random, it is stochastic. It's a stochastic phenomenon. If it weren't, we should have seen a little peek in detail saying that there is some anomalous activity falling through Earth in some moment of the year. So we managed to publish this. We went through all the data one by one.
Ya su mamá también. La
One
saludé.
register by one.
También.
And there were over more than 24,000 registers to look at. Some of them didn't contain any information useful to us, but they are coming more and more information as we speak. So the other aspect of meteorite falls is, of course, the delivery of materials from outer space to Earth. And we know that this delivery of material was probably very important to the origin of life. In particular, we were interested in the delivery of minor bodies of the solar system, such as meteorites, interplanetary dust particles, comets, and asteroids. We tried to study the amount of total mass of carbon. We know that carbon is the most important element for life. So we tried to calculate how much mass of carbon was delivered during the formation of the Earth and the first eon of the Earth, the first billion years of the Earth formation, which is the Hadean eon. So in that sense, we know that the organic matter have some difficulties to get established in a rocky body in the solar system. It cannot survive high temperatures, high impact velocities, but if the planet had a thick atmosphere, it might allow for a small rate of deposition for these small fragments. We also know that due to other papers that simulated heated conditions can allow for the synthesis of organic molecules. So this creates a sort of paradox with Chiba and collaborators called the Janus paradox, Janus being the Roman god of endings and beginnings, because the high temperatures and impact velocities can sterilize the surface of the Earth and atomize organic matter, but it can also catalyze the synthesis of biological relevant organic molecules. So we are standing in a point of where do we start? So in our work, we decided to assess how much mass of carbon was delivered during the first billion years of the Earth. We used this model based on Chiba and Carl Sagan, developed in the 90s. And we tried to find the present rate of fall in kilograms of carbon per year for 3 different populations of minor bodies of the solar system, such as asteroids, comets, meteorites, carbonaceous chondrites, in fact, and interplanetary dust particles. And what the data show us is that the total mass of carbon in this iterated in these first billion years of the Earth is of the order of 10 to the 16th power of kilograms of carbon, which is a huge amount. Just to bear in mind, this is more or less the total amount of active carbon at the moment, in the present. I mean by active that the carbon, the element, can enter any cycle, not just a biological cycle, any cycle of the Earth is not storage somewhere. And to bear in mind that the total biomass of carbon in the present is 2 orders of magnitude less. So the amount of carbon delivered during the Hadean, during the first billion years of the Earth, could explain the emergence of life. Of course, this model have some important biases. This model considers the Earth as a whole rocky surface. There is no oceans in these models. There's no consideration of endogenous synthesis, such as hydrothermal vents or destruction by impacts, outgassing, for example. And of course, we are not considering the other important elements for prebiotic life, such as oxygen, nitrogen, phosphorus, sulfur, and metals. But if some portion of this carbon was lost during the first billion years, the available carbon should be enough to start life. So this is an ongoing work. If you have any comments, we could talk about it later. So moving on, I started last year my master's degree in biology, which I also emphasized that this master program is also free. This is an STEM program for master and doctorate, which is free for anyone interested in. And as a result of this astrobiology related curricula that I am pursuing, I try to understand how life could have come together. And of course, we know that life is in itself a self-sustainable entity, and its building blocks should have arrived as an autocatalytic network. But of course, if they are diluted in the ocean, reactions cannot occur because molecules cannot find each other. So we need some sort of vesicle, a compartment that can allow for reactions to occur. This, of course, creates some some problems, some technicalities that we should, which I am interested in. For example, the osmotic growth caused by internal reactions. So what happens if the cell or at least the protomembranes self-assemble in a hydrothermal vent, for example, or any other environment. We know that this self-assembly is thermodynamically stable, and it can allow for the influx of molecules that are permeant of this membrane. But if a reaction takes place inside the membrane and creates some product that it is not permeant to the membrane, the compensation implies that water should come inside the membrane, and that could generate a burst of the membrane, a rupture of the membrane, releasing all its content to a now enriched environment of these new molecules, which can then be recycled in a new process. So this is an ongoing work. This is purely theoretical. We are trying to find out how the dynamics work, if the autocatalytic networks can be sustained in time, can persist inside the membranes for a long time. We could have a moment where there were some sort of population, which can, of course, be subject to some certain of evolution, and therefore evolve towards the life as we know it. But of course, if the velocity at which these membranes burst is very rapidly, we cannot have this type of life, so we have to find another mechanisms. We currently don't have the possibility to do experiments. We certainly would like to, if we don't have the equipment nor the knowledge of how to create artificial membranes. But if you are interested in collaborate, do email me, do talk to me, and we can maybe have some sort of collaboration. So moving on to the last part of this speak, I am a high school teacher as well, a science school teacher. In particular, I am interested in astronomy and physics. And we have, 2 years ago, we started a project about how to communicate science and how to study the universe beyond what our common senses can tell us. So here's a picture of the Helix Nebula from different views, from different light filters, I should say. For example, here at the top left is a visible light photograph. I think bottom left is X-ray, and bottom right is gamma ray, and top right is infrared. So the same object of the universe seen in different lenses can tell us about different things. For example, in visible light or infrared, we can study the composition of the nebula. In gamma rays, we can study the white dwarf in the center, etc., etc. But moving on that idea, we try to answer the question of how blind people can study astronomy, how blind people can study astronomy. And we come up with the idea that that given that light and sound are both waves, they share some properties such as frequency, longitude, etc. we can translate some of the information into one into the other. So we come up with a sort of scale. We know that stars have some color. We can study the index color. We can classify them by color. We can study the color of maximum emission, the longitude of maximum emission. And we can also try to generate some sort of translation between light and sound. If you are familiar with the musical notation, you would recognize that each tone can be correlated with some color. And we generated a sort of device that would allow the blind people to differentiate different types of stars accordingly. So, for example, at high school level, you can purchase some of these microchips, which can easily be programmed to correlate some music with light. So, for example, here is the device. See, this is the microchip. This is the battery. This is the headphones. There's a lot of cables. This is one of my students testing it. So the project consists as follows. You have a constellation. The students choose a constellation, and they developed some circuitry that is connected to headphones and connected to LED lights. Each LED had a color corresponding to the actual color of the star, and as they invite someone, they blindfolded it, and then the blind person, I don't know if the chat is working properly, but okay, sorry. So as the blind person tries to discover the layout of the constellation, they come across with the button. If they press that button, they would hear the sound, a sound that should correspond in tone with the color of the star, but not also with the tone, but with the volume as well. Volume in this context represent brightness of the star. So, for example, if you come across a star, you press the button generates the sound. If the sound is too high pitch, it would be a bright star. And it should be a blue LED corresponding to it. If the star is dimmer, you should have a red light corresponding to it, and a lower pitch. So this, we managed to publish this a few years ago. There's plenty room for improvement. This is very homemade. This is very sort of experimentation. We certainly would like to have some feedback from you. We certainly want to have some feedback from actual blind people, because we didn't have at that time blind people to use as test subjects. But if you are interested in knowing more, please do contact me. So I would like to thank you very much all for the invitation, and we'll be happy to answer your questions.
So hello again to everyone. My name is KARINA ORTIZ LEYRA. I'm an earth scientist specialized in space scientist, space science, sorry. And let's begin with the presentation. So, Titan is Saturn's largest moon, comparable in size to Mercury. It's of great interest because its dynamics of the synthesis of molecular organic molecules. We know this based on our knowledge of early Earth. For example, the Urey-Miller experiment discovered the formation of amino acids. Since Titan shares similar atmospheric conditions, we classified it as prebiotic laboratory that mimics the environment of early Earth. In comparison, early Earth and Titan shares many similar features, as a stable chemical cycle, the presence of atmospheric acetonitrile, the formation of amino acids, a carbonated surface, and the reception of ultraviolet radiation. When I start this research, I synthesize 4 principles. 1, molecular evolution. 2, the formation of organic compounds. 3, the physical-chemical dynamic of solids, which are clusters of organic compounds on Titan atmosphere, and the similarity in prebiotic formation pathways. The methodology uses the simplest amino acid, who is glycine, in oligomers 1 through 6. We use calcium carbonate to simulate the surface and acetonitrile as promoter. And we use computational simulations, polarized light microscopy, DSCA, and FTIR spectroscopy. For these last 2, we test samples both with and without UV irradiation. So, simulations allow us to predict molecular behavior. This helps us verify the compounds before the experiments, which saves materials and reduces waste, something vital for our planet today. We obtain data and potential energy and reactivity. Here, we model a calcite surface with its characteristic trigonal structure and add it to glycine 6 to test its reactivity. An important finding was the formation of beta sheets in glycine 6. This structure is the basis of molecular machines, and this is a crucial indicator that the system will show catalytic properties in our results. This technique of polarized light microscopy show how light travels through the mineral, highlighting the cohesion between the mineral and the amino acid. We use 10x objective and a camera, this one, which is the blue visible as the double lines. This is the big refrigerance of the island spot. The refrigerance is visible as double lines here and here under the mineral. And this is the sample that we use on the lab. Oh, sorry. Okay. So we can visualize. Oh, sorry. Let me move this. Okay. We can visualize the equation. Glycine has a round edge and high opacity, while the mineral is angular and transparent. So glycine 1 shows cohesion with the carbonate, but in glycine 6, it adheres strongly to the surface. This provides that the larger it's the oligomer, the better the cohesion with the mineral. DSCA allows to measure energy change in the system. we use a heating range to 20 degrees per minute, a heating range from 30 to 1500 Celsius, and I use alumina crucibles. These give us thermograms showing the thermal energy and the transitions on the system. In the results of DSC without UV up there, I want to highlight how the system stabilizes when acetonitride is at regulating the endotherms. And under UV irradiation, the temperatures show no abrupt changes. But the system with acetonitride become very irregular. This confirms our simulation that the higher molecular weight on glycines are more unstable and more likely to react with the environment. And for FTR, this FTR allows us to visualize molecular by reading vibrational spectra. We use a range for 4,000 to 650, and we analyzed 4 scans per sample, and the result of the spectrograms were analyzed by extrapolating the data into software. I use Origins for this data analyzed. And I like to highlight the sorry, on this line. This shows the NH stretching for hydrogen bonds. So these bands between 1700, sorry, and 1500 represents the amides 1 and 2. So comparing the samples, these bands are longer in lysine 5 on green and lysine 6 on blue, supporting the idea that the largest oligomers are more likely to show molecular evolution. And this is so visible here and here. Okay. So in this table, I show molecular matches. So here we have glycine 5 that match the secondary aliphatic amide found on alanine, and lysine 6 that shows an amide with carbonyl, and it's also present on alanine. But for a visual comparison, here is the alanine on pink or red, something like this, versus our system under UVC, including glycine 6, carbonate, acetonitride, and UV irradiation. So the shifts in the band shows a very high possibility that the system evolves into more complex molecules. So, in conclusion, number 1, simulations suggest that abiotic peptide synthesis could lead a catalytic properties. Second one is that we see molecular diversity in the amide bands. And number 3, calcium carbonate as a substrate that favors synthesis with acetonitride as a catalyst. And last one is that the UV irradiation increased reactivity and could be for new amino acid on the early Earth and possibility titan, sorry. So glycines with high molecular weight under UV irradiation provide a better scenario for prebiotic synthesis. So we can see the titan evolution similarly as the early Earth. And I know that is so fast, but I'm so nervous, honestly. So here are my references. I know that the list is so long, but feel free to take a screenshot or I can share it with you later. Here you can also find my 2 published articles, here and here. And well, finally, I want to thank Norcells for the invitation, this forum for your attention. Thanks to my tutor, of course. And here is Sohan, and here is Miri. So thank you very much.
Very good. Don't worry. Take it easy. It was a huge amount of work for you as an undergraduate student. This was a great amount of work for a person who is not even in master's. So congratulations. As I told you, your work is very interesting. Actually, yes, you are receiving a lot of claps. A lot of
Thank you so
claps
much.
of reaction.
Hello everyone, my name is ERICK de CARVALHO. I'm from Federal Institute of Ceará in Brazil. Today I will present my topic of research is circumstellar habitable zone and ultraviolet radiation. Next slide.
Here or the next?
First, a brief introduction about myself. I'm a biologist. Actually, I'm researching this for a long time. I'm in extension scholarship, youth apprentice in AI, and I am finalist of IAC, is International Astronomy and Astrophysics Competition. My research interest is radioactive equilibrium, temperature, planetary geology, habitable zone, and exoplanet habitability. During my graduate, I have produced an algorithm that simulates a system, the system of Tardigrades star, and using Python simulations, we could calculate the habitable zone circumstellar, the circumstellar habitable zone. And my undergraduate thesis is about that. It's habitability analysis and radiative equilibrium temperature of the Tardigrades star system through the Python simulations. Application in astrobiology is under supervision of Professor Francisco Emerson. Next. Here is a summary of my presentation, and I will talk about exoplanets, the first exoplanets and the discoveries, the detection methods, how to find habitable exoplanets and zones, circumstellar habitable zones and ultraviolet, biosignatures and change of lives, how to find lives of the universe, and atmosphere in modeling. Is here I will show my results of my thesis. Next. First of all, I will talk about exoplanets. Exoplanets will, next slide. Exoplanets, the study of exoplanets is very recent and very important. In 2009, Michael Mello and Didier Queloz received the Nobel Prize in Physics. They were recognized for discovering the first exoplanet orbiting the solar type star in 1995. I don't know what to say, but is it? This thing is discovered through the planetary transit, and this discovery is open this horizons to research in astrobiology and astronomy. Since the discovery of exoplanets, the number of discoveries has grown fast. Today, we have 6,000 approximately confirmed exoplanets. As you see in the chart, there are many type gas giants, super Earth, and terrestrial planets. Next. And here this is a graphic of this shows the habitable zone on the IX. We have a temperature of the star in the x-axis. We have the amount of light the planet receives and then the green area is the optimistic habitable zone, and then the blue are the conservative habitable zone, where the liquid water is likely to exist in the surface. Next. Our solar system have an habitable zone too, and we are inserted in this zone. As you can see, our solar system is the right in the middle of the green zone, and Mars is in the edge. Venus is too hot, and the planets are too cold to have water and to have water liquid. Next. Here as stars classified by spectral type and temperature. Hot stars are type O or B. Cooler stars like Hedgehoff are type M, and the Sun is G-type star, which the surface temperature is around 5,000 Kelvin.
5,000.
Next. Yes. 5,000. And this is subject we astrobiologists Mars research. This is the stars and the terrestrial planet Earth light. Here is our diagram, the Hertzsprung-Russell diagram. It plots the luminosity of the stars against the temperature. Most stars, including Sun, are in the main sequence, which is the first star of the life. Is such incredible because our star, Sun, is in the middle of this graph. Next. Here we are talking about the detection methods. The detection methods is there are many ways to find exoplanets. However, two methods are responsible for the most discoveries: radial velocity and transit. Radial velocity and transit is the two most most researches in the astronomy and in the astrophysics. And we astrobiology catch this data. In this data, we catch in insert in calculations, in models, mathematics, and in this, sorry, my bad, my survey. And
Don't worry.
here's some ways to find the exoplanets. Next. Radial velocity. Radial velocity measures the minimum mass. Radial velocity measures the room of the star is given to the minimum mass of the planet. The planetary transit, we observe the shape of the planet when it passes in front of the star. It gives us the radius of the planet. Combining this, we can determine if the planet is rocky or gaseous. And in the
in the bottom. Mm-hmm.
In the bottom, planets with mass
Under
10 Earth masses or under 2 radius Earth are considered limits to search these planets and classify it as rocky planets. Next, we are talking about now about biosignature. The biosignatures is the life, is basically life. Life requires complex chemistry. We have detected many molecules in space, such amino acids, PAAs, and other precursors of RNA. These are the building blocks of life. The chunks. This is the key. The key of life is. Next.
Okay, yeah. Sorry.
And in their spectrum, if the alien zone means the alien located for the Earth, this is what we will see. The spectrums show absorption lines for water, ozone, carbon dioxide, and methane. Together, these gases are strong indicators of life or biosignatures. Next. The periodic table for astrobiology, we look for these elements. These elements are essential for the life. The most important here is the carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, or chunks. Next.
Exactly.
In the next section, we are talking about my research in the atmosphere and modeling, physical modeling of this habitable zone and equilibrium temperature and various calculations. Next. Here is the such formulas, is equations. And these equations is about the coefficient of polynomial equation that fit the stellar flux based on the temperature difference between the star and sun. To calculate habitable zone, we accurately use these mathematical models, and we consider the runaway graphs. The slide. Yes.
Sorry.
We consider the runaway greenhouse, the effects, the inner edge, and the maximum greenhouse, the out edge, This equation depends of the star effective temperature. And next slide. Here is the table of the work of Kaparapu et al. It provides a coexistence we use in our Python simulation. My research is in this program, in this algorithm, which simulates different types of Earth and Earth can, because my English is these boundaries for different planets. Next. Besides the temperature, we must consider the ultraviolet radiation. The high levels of UV can be dangerous for life, but the low levels might be necessary to origin of life. This chart correlates the UV luminosity with the temperature of the star. It's from Shelley et al. 2004. 2004. Next slide. In the next slide, we are talking about these models, and I applicate these models in Python and in Color. We can apply to in Mathematica. Here, row from Mathematica, these models, and relate these two. I applicate for the Tea-Garden star system. Here is a red dot to confirm planets, Tea-Garden B and Tea-Garden C. Next slide. And in this slide, we can see the specific system. In these slides, we can see the programs we can use in this. Next. This is the Tea-Garden star comparing in the red. Next. And this is the models we calculate in algorithm. And the ALBERTO limit for Tea-Garden search luminosity is 0.0072, and the temperature, the effective temperature is 3.550 Kelvin. The recent value is in 0.00219, greenhouse in 0.0278, and the maximum greenhouse is 0.0540, and the elements is in 0.0569. Next. This chart is the result of my Python simulation. I implemented the Kaparapu model to plot the habitat zone of Tea-Garden star. The dark green is the area of the conservative habitat zone. The light green is the optimistic. As you can see, Tea-Garden B, the first dot, is the right in the edge of optimistic zone. The Tea-Garden C, the middle dot, is constantly in the conservative habitat zone. And Tea-Garden D, which is the planet is not confirmed, is in outside of the two habitat zones. Next slide. Depending on the composition, these planets could be rocky worlds, ocean worlds, Hesian, or mini Neptune. In this case, it's rocky. Next. Why studies? Great conditions for life, possibility of extraterrestrial life, understanding on our planet, and the limits of life. So, why we study this? We want to discover the conditions of life and understand the limits of biology, the physics, and the chemistry of life. And we have the question, we are alone? Next. These are subjects of the studies of in the astrobiology, and my work is in exoplanets and in stars and in various of these topics. Next. T-Garden is not the only one. There are many potentially habitable worlds near, like Proxima Centauri B and the TRAPPIST-1 planets. Here, these charts of the PHL are visible, and we have various planets in the habitable zone. Next. And finally, I want to show you this image. It shows the Earth seen from Saturn. We are just that tiny pixel that reminds us the famous words of Carl Sagan about the pale blue dot. We say everyone we love, everyone we know, and every human being who ever was life of their lives is the mode of the dust. In astrobiology, we look to life everywhere. But this perspective teaches something important. The Earth is our only home, and we must cherish and preserve it. Next. Here is bibliography. We have a book of the astrobiology in Brazil. It's Astrobiologia, uma ciência emergente. It's a book in Portuguese, but we have two in English. The Exoplanet Atmosphere by Sarah Seger and the Handbook of Astrobiology by Verkov. Next. Thank you for the conversation and thank you very much for the attention. If you have any questions, feel free to contact me via LinkedIn or maybe I'm disponible. Thank you.
My name is Luis Alberto ALLCCAHUAMAN-HUAYA. I am from Peru. I live in Lima. I am a biologist in Peru. I study Cayetano Heredia University. I member Biomed, Bioinorganic Chemistry in Medicine in the Biomed, National University Engineer, Peru. And I am founder of Proyectos Tardigrades Peru. Well, you are observing tardigrade isolated from most in Peru. You see a tardigrade movement. No? You see eyes, your body, clouds, and other characteristics in the fantastic animals. There are morphological characteristics tardigrade, for example, rounded and segmented body. Tardigrades have digestive, nervous, and reproductive systems. This These are very spectacular. Tardigrades have legs with claws, and
Yes.
tardigrade has eyes. And
Yes.
size with the range. This uh, this micrograph taken by me. This belong macrobiotics genus. Characterbiosis. Characterbiosis is is amazing state that is a metabolic state. It means suspension of psychological functions. In this video, you watch a tardigrade belong Melanesium from Ayacucho in Peru. This video recorded by me. This
Oh,
very interesting animal is very teeny by microscopy. You see, and character biases is stay very interesting. You see in this picture uh, unusual form that are degraded. Type of biases for ways, for example, cryobiosis and anhydrobiosis, osmobiosis, and axiobiosis. Uh, w- why is important cryobiosis? Well, cryobiosis is very interesting uh, to scientists. For example, tardigrade has interest in several molecular, for example, protein. No? DSP protein is very interesting because this protein protects DNA tardigrade. Well, I astrobiology investigation involving tardigrades. For example, there's a project you this work carry on in 2006 uh, by European Space Agency. Well, you see the results. There's a project when survival microbiotics research specimens. Um, you see uh, high high survival percent uh, and expression protein EHSP head shock protein. Well, when the two protein is very high, when tardigrade exposed in space. Other project name is Tardigrades. And in this project, you used other specific tardigrades, for example, Paramacrobiotis richardsi, Ramazotius overhauseri, when uh, when in comparison two proteins, glutathione and glutathione peroxidase, because this antioxidant protein confers protection tardigrade. Other project is TARDIS project, when tardigrades can survive, no, in space in two species, Richard's corinifer y Melanesian tardigrade. You see uh, two spatial parameters, for example, UV, AB, and um, space vacuum from space, no? And the result that the survives. That's very fantastic. Other project, Rotaret, Rot- where researchers conifer survived after export to orbit space. The result, the survival was higher under low dose cosmic radiation conditions. In Peru, Peruvian astrobiology associations carry on uh, experiment when tardigrade, macrobiotus, send a statopher using statospheric value. The result is 42 number of live tardigrade in Petri dish number. No? And you see macrobiotic characteristic by microscopic. This is the first stratospheric tardigrade survival study conducted in Peru. Now, Martian astrobiology simulates experiment involving tardigrades. For example, um, comparison to situation, Mars and texture camber. Texture camber is simulated Martian conditions. You see environment parameters in this table. And right in tardigrade, you said Ramazotius variernatus, no? In survival percent under different conditions. This tardigrade survived in two conditions. You see survivor, the tardigrade in two conditions, and different magnesium perchlorate concentration versus control. And another in this another picture, comparative analysis body length paramacrometer experimentalis under condition and exposure to different magnesium perchlorate concentration. The result is tardigrades survivor in different concentration en magnesium perclorato. Why? Magnesium perclorato was discovered in Mars. You see schematic illustration cellular and molecular mechanics, potentially involved in epithelial transport. This is a hypothesis in tardigrade. You you see several proteins, for example, el D2, small head shock proteins, and antioxidants, for example, superoxide dismutasa, glutathione peroxidase y catalase, ¿no? La application in biomedicine using tardigrade. For example, el D-use, D-sub, is very interesting because protect protects DNA in in
It
future
protects.
in future application, for example, in medicine against cancer. No? This is a very special word. Tardigrade resists against radiation because DC protein protects tardigrade from mm, radiation, and DC protein combine RNA y using nanoparticles direct injection in tumor against cancer y, durante radiation therapy, ¿no? That result de su protect no de not que no de tumors cells. ¿No? Other application the tardigrade de su protein against ultraviolet into the cellular dimension in plants, for example, and insert DC protein in the plant against ultraviolet and protect the plant development. Conclusion, Tardigrade exhibit extraordinary survival under extreme condition. Thanks, thanking space exposure, cosmic and UV radiation, and other parameters. And the first stratospheric balloon experiment in Peru confirmed the ability of near space biological research as an intermediate platform between laboratory and orbital study, Martian simulation experiment for the support, the relevant or tardigrade, and discover protective protein, for example, this sub, a highly strong potassium for radiotherapy application in agriculture. No? Finally, you see 4 pictures tardigrade taken by microscopy. No? And you see different tardigrade types, for example, microbiotus, Echisnicus, Mesobiotus. It's very interesting for by me and other um, investigators. No? Thanks a lot.